The protocol aims to simulate in vivo conditions of intervertebral disc degeneration to investigate its pathophysiology without the need for animal models.
Compared with in vitro cell culture, the bioreactor organ culture technique maintains the cells in their native biological and biomechanical microenvironment, in addition to the supply of a controllable and reproducible culture condition.
Start with rinsing the entire bovine tail thoroughly with tap water to remove dirt and hair on the surface. Then immerse the tail in 1% betadine solution for 10 minutes to disinfect the surface. Use a scalpel number 20 to remove the soft tissue from the caudal spine to facilitate the identification of the intervertebral discs, or IVDs. Remove the spinous and transverse processes of the vertebrae with a bone removal plier. Cut transversely with bone pliers through the middle of each vertebral body to obtain individual motion segments. Then put the collected motion segments in a petri dish with a gauze wetted in Ringer's solution. Locate the IVD in vertebra by moving the motion segments gently. Then identify the location of the growth plate by touching and finding the convex side of the bony end plate. Cool the blade of the bandsaw with Ringer solution and use it to make 2 parallel cuts in the growth plate of the IVDs, one on each side. Transfer the IVDs to a clean petri dish with clean gauze wetted with Ringer's solution. Scrape off the vertebral body and growth plate using the scalpel blade, leaving the end plate intact. Position the 2 surfaces flat and parallel for the loading procedure and transfer scraped IVDs